Morning, people. I hope you're doing well, my dear lovelies, on the blurriest stream of the year. Daylight is saved, and yet, despite the inevitable and expected sleepies, I've brought you something. It's not the caffeine, that's for me. But what I brought you may contain science. Alright, so I bring a lot of bioscience. I bring the occasional science about science. And today's story has a little bit of both, as a new study in current biology investigates neuroscience performed in male versus male and female lab animal populations, and whether there is any basis for scientists' assumptions about how the sex of an animal influences the consistency of data about their physiology, and by extension, how scientists should handle that in biomedical, and again, in this case specifically neuroscience studies. Medical science is not great with matters of sex and gender, evidence for which is available throughout the history of global science. But 20th and 21st century medical science have more specific methodological issues. In mammals, sex has a significant impact on systems throughout the body, including in all sorts of places you wouldn't expect it. It comes up in like liver or kidney studies or something, and it's like, what are reproductive hormones even doing here? Go back to class. And it turns out, well, sex influences our body in countless ways, and that's one of the reasons it's super great to get people on the hormones that are right for them. But I digress. Unfortunately, these differences are inconvenient when you're a scientist trying to isolate your variables. If your results are all over the place, is it due to the phenomenon you're studying, or is it due to some unexpected interaction with sex hormones? And any student of medical science history can guess which sex was made the default in animal studies. Now, lab rodents don't have the same cyclical estrogen pattern as humans because mice, well, they reproduce very differently. But lab mice do go into what's called estrus on a regular basis, and there has been a persistent concern in medical science that lab mice in different parts of their estrus cycle will produce different data that would add noise and detract from the statistical significance of the results. Not only might female mice produce different results than male mice for a given experiment, but the idea is that a female mouse would produce different results in 1 week than that same mouse would produce in a different week due to the experiment's timing against the mouse's estrus cycle. And obviously for some studies that is going to be true. If you are looking at estrogen or progesterone levels, yeah, guess what? They're going to be influenced by the mouse's estrus cycle. And then for other studies, it's not going to be true, because female mice are not completely physiologically transforming week to week. Mammals are not frickin' xenomorphs. The hard part is figuring out which is which, because if we knew enough about a given phenomenon to know all the biological factors involved, we wouldn't have to do fresh science on it, right? So this is a genuinely difficult methodological problem for bioscience. These are non-trivial differences. That's why gender-affirming care is necessary medical care. But I digress. And they significantly increase the complexity of experimental design in order to even try to account for them. Historically, biomedical science as a discipline has not taken the most productive approach in response. In many, many fields, researchers have used solely male lab animal populations, intending to eliminate the variability issue introduced by female animals' reproductive cycle. Put bluntly, the idea is that you're just using the consistent ones. The consistent ones in your population to try to study whatever physiological phenomenon you're studying without the confounding factors of the sex with more individual variability. And pile enough of this kind of study on top of one another, and you have not built enough bad ideas into a good idea. Now, your whole body of literature in a given subfield is using single-sex populations in their studies, and what? Are you going to go back and replicate all of that in female lab animals? Nah. we'll just use the male and female animals in a future study. Then you get to that future study and, well, the reviewer will ding me for not having the same protocol as my previous study. I'll just use male animals again. It'll be easier. And by the time this science hits humans, we've ended up with pharmaceuticals that hit the trial or even approval stage only to show surprising effects on estrogen-dominant people. Like that one time when they found out a whole bunch of folks were taking too much Ambien because the dosages weren't properly tested for everyone. Or you end up with diagnostics that don't work for a huge swath of folks. Or you even just have basic science about the human body that may or may not be applicable to everyone because no one ever bothered to look. By taking a shortcut on our methodology to avoid one kind of problem, a lot of fields slowly grew this deep methodological rot that represents a much more profound problem. And neuroscience and behavioral studies are among these fields where the literature has a lot of single-sex male lab animal population inertia. And our study today is from this discipline, as they investigate patterns of open field exploration among male and female mice. The way mice explore an open area is apparently a stereotyped pattern to the point that scientists can accurately predict what drug a mouse has received and the dosage of that drug for certain kinds of pharmaceuticals based on changes to the mouse's exploration pattern. Kind of like how toxoplasmosis makes mice more bold. It's a similar principle. It's a change to the exploration pattern. And these patterns change with all sorts of changes to inputs, like sensory cues or whether the mouse is hungry or in pain. So it's entirely possible that this is the sort of thing that would vary between male and female mice, and that if murine exploration patterns change in response to cues like hunger, it's certainly at least intuitive that a female mouse's estrous cycle might be one of those kinds of factors. Spoilers. It's not. Differences within male and female mice were much more significant than between male and female mice, and female estrous cycles were effectively negligible factors. In fact, male mice demonstrated greater individual variability than the female mice did. So in this case, an all-male mouse population for consistency would have had the opposite effect. So it's a cool study, it's got the counterintuitive result, but okay, I almost feel mean saying this, but why is this in current biology? Female mice show lower individual variability in exploration behaviors than male mice is a perfectly cool, interesting finding. But it sounds like more of a Journal of Minor Murine Behavior article than what I might expect from a high-impact journal. So, if I might advance an opinion, part of the reason I suspect this finding might have made it into current biology is on the back of its sexy methodology. It actually is this really cool machine learning and computer vision stuff. Over the longitudinal study, the unsupervised machine learning broke down the movements of mice into what the authors call syllables of exploration behavior. Pausing, stepping, these atomistic behaviors, really. By following each mouse over time and breaking down their exploration behaviors down to these itty-bitty bits, scientists could measure the degree of behavioral variation for each mouse, and in so doing, not only determine how variable an individual mouse's behaviors were, but because it is a longitudinal study, they could see whether variability in female mice correlated with their estrous cycle. So, studies with machine learning, everybody's favorite child right now, are excellent stonks at high-impact journals for the moment, although I do find the whole design very nicely put together. It's not just that it's using the sexy tool. I think the experimental design's pretty nice, but I'm not really here about the method I've built today's whole scientific soapbox upon. No, that's the study's findings, isn't it? And what they say about how we do science. This study is from a field that long took up a rotten methodological bargain in the name of scare quotes consistency, and I should say, even if it's imperfectly enforced, institutions like the National Institutes of Health and the FDA, as well as parallel organizations around the world, have, for years, required that publicly funded science use animals of both sexes without a compelling methodological justification for why they couldn't. But, if we're being honest, a lot of bioscientists see it as a worthy goal, but a mildly compromising pain in the ass. A potential introduction of noise to the data and frustrating confounding factors. What this study reminds us is that often enough, that mildly compromising pain in the ass thing isn't the case at all. And the intuitive assumptions we might make, that cyclic hormones equals more consistent, may not only be untrue, but completely the opposite. That in attempting to reduce complexity in our experimental design, making assumptions and taking shortcuts to avoid the methodological problem we think we see, only damages the integrity of our scientific result and creates more complications down the line. No, this study absolutely does not prove anything universal about mice or behavior or variability between specimens or populations. On their own, frankly, these findings are not world-shattering, but they do make you wonder what we would have found across all sorts of studies if we hadn't messed up our experimental design by preventative optimization. And by refusing to front-load the complexity of our bioscience and contend with potential confounding factors directly, we may have ended up with a much poorer understanding of the overarching governing mechanisms by which sex hormones influence physiology overall. We're all worse off for that, especially cis women, trans and non-binary people, and intersex people. There is a tremendous amount we don't understand about the interplay of hormone profile and health. I can confirm, trans people have a zillion questions, like, anecdotally, a lot of us say that HRT does this and this and this, but there are no peer-reviewed studies to establish it more definitively, and yes, that's because there's very little trans or intersex-specific medical research, but it's even more fundamental than that. In the name of making things easier and more consistent by doing so many studies with single-sex populations, we've accrued this huge scientific debt that could have provided a foundation for investigations into trans and intersex-specific medical studies. Look, I know I cover biology a lot. It's no secret it's my favorite. And I feel like every time I do, sooner or later I say something along the lines of, biology is complex. Well, guess what? It is. There are a zillion things going on, all at once. And you never know for sure if you've accounted for every factor that's in play, or that those factors are behaving in the way you'd expect. A lot of good science is backing out and asking yourself, wait, why do I think this is the case? And what if I'm wrong? And today's mouse study is just a tiny microcosm of why that's so important. It's not a study that blows everything wide open. If you happen to already be doing science with machine learning, you're living large right now. You should absolutely shoot for high-impact journals, and that may be part of this paper's story. But it's another brick in the wall of an ever-growing body of literature indicating that single-sex study populations aren't cleaner science, they're worse science. And NIH and FDA and parallel requirements are not watering science down or demanding unreasonably large, expensive study populations. No, they're making our work stronger. Because yes, experimental design requires that we make some assumptions sometimes. But female mice are more inconsistent than male mice was always a huge assumption based on incomplete evidence that's not true a whole bunch of the time. And making that assumption ultimately causes problems for both science and societal equity, and all of that together adds up to some freaking hubris. But Dana Levy lead author of this study and research fellow in neurobiology at Harvard Medical, put it pretty well. Assumptions that affect the way we conduct and design our science are sometimes just assumptions, and it is important to directly test them, because sometimes they're not true.